This is the Kratom Science Journal Club with Dr. Jonathan Cachat, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. Okay, so the article that we're going to talk about this time is some observations on the pharmacology of Mitragynine. It's from the International Archives of Pharmacodynamics and Therapie from 1972.
72?
Yeah, way before even I was born. It's from SmithKline and French Laboratories out of Philadelphia. And this company originally started as a drugstore in 1830. By 1929, it was a research laboratory. One of its chemists, Gordon A. Alice, got a patent for amphetamine in 1932, and they merged to form SmithKline Beecham in 1989, and they merged with Glaxo Wellcome in 2000 to become Glaxo SmithKline.
Gotcha. Yeah, I'm glad you laid that out, because I didn't necessarily realize that these were R&D divisions for ostensibly pharmaceutical companies or private companies. These researchers weren't coming out of a university. It's just interesting to note that, though. I don't think that there's any difference in how you can assess. This is early product development. It's the same type of knowledge. You can't just sort of write it off because that. And a lot of work, a lot of good research gets done in those settings. But just interesting to note, this is an early R&D in the pharmaceutical company, which I think, in light of where we go through this what they report, it's just sort of interesting to know, has Big Pharma known about this the whole time? Like, that's an interesting question.
Yeah, and I have that. I have that as one of the questions, because it was like way back then, and they were drawing a lot of the conclusions that we have seen in more recent studies. And then I looked up a little bit about the guys. There wasn't much about Edward Macko, the main chemist there, but the other guy, Dr. Weisbach, Jerry Weisbach, led the team that discovered and developed atorvastatin, or Lipitor. So,
whoa, dude, that is a big one.
Yeah,
I mean, that's a huge one.
2002. Yeah, he's from.
Wow.
This is from University of Michigan, and he was like a visiting professor there after that was kind of his retired job, and then the other guy became an executive of the company, Dr. Bryce Douglas. He was a Scottish chemist, but yeah, the Lipitor guy. That's what I thought was pretty interesting.
That's some heavy hitters that then went on to, set the found. I mean, torvastatins, or statins in general, are one of the most effective medications we have, and are, one of the only truly, when you look at the data over all the years we've used it, it's one of the only true pharmaceutical interventions that regularly, confidently, statistically significantly shows it reduces the ultimate incident of heart disease or heart attack.
Yeah.
So, even there's cholesterol is a whole other thing, but statins revolutionized the ability to treat that, and there hasn't really been any fallout. So, yeah, these guys definitely look to be curious and performing a battery of tests, this time, of course, on Mitragynine, in animal models to start distinguishing broad behavioral characteristics.
Yeah, so they did a lot of weird stuff to animals for this study. I had a list. Sure. So dogs were exposed to radiant heat almost to the point of burning their skin. They injected brewer's yeast into rats' paws to induce inflammation. Then they kind of jabbed them with a needle. This is all like pain tests. They put mice on a hot plate. They put an iron plug in a dog's trachea and used a magnet to induce coughing.
Let me pause you there. So you're good. So we're going into a different approach too, and I'm glad you're running through all this. But yeah. I just want to do a slight distinction of clarification.
Okay.
The hot plate test is essentially, you get a control on there, you turn up the hot plate, and at some point the animal will get their feet off of the hot plate because it comes to unbearable. Then you give them something that's believed to be an analgesic. The hypothesis is that they stay on that plate longer or can tolerate higher temperatures. It's the same with the tail flick test and with the brewer's yeast inducing inflammation. So all three of these tests are fairly common, not necessarily done in the same exact way that they did in this paper, but are all designed to gauge the analgesic potential of a treatment, right? Okay.
Yeah. And then the other thing is they fed rats charcoal, which that's not that bad, but they did a cardiovascular test on a dog where they stuck a tube down its throat and then cut off its air. They got a monkey hooked on morphine, which is pretty funny. Junkie monkey. And then they gave... The part that I didn't like is they gave 12 beagles were given mitragynine over 93 days, and then towards the end of that description, they said they were sacrificing an autopsy. It's like, oh, did they have to die? Come on.
Yeah. We don't want to get the podcast canceled for talking about
did the doggies go to heaven? That's what I want to know.
They definitely went to heaven.
Okay, good. Good.
And no one is operating on donkeys nowadays.
I didn't think so.
Okay, let's push that to the side. Let's talk just a little bit about, so at first we were talking about analgesical behavioral response. Okay. Do these compounds seemingly increase the threshold to pain? So they affect it at reducing pain. When we went to the charcoal one, it's kind of interesting because another well-known and well-documented property of opiates is that they can lead to constipation. The smooth muscle in your intestines and in your internal structures, when exposed to opiates, start moving more slowly. So I'd never heard of this charcoal maneuver, but they were essentially putting charcoal into the animal's body and euthanizing the animal, and then seeing how far that charcoal had made it through its intestine. And the idea being, with codeine or the standard opiates, it's maybe 50% more or less traveled than the control. They wanted to see where mitragynine landed there. So can it inhibit smooth muscles in the digestive system? And I want to take a small note here just to mention, sort of like how we were talking about the operant conditioning, and all of these tests are pretty straightforward. The one that we went over with Dr. Citation, a little bit more complicated, involving like conscious and memory. Most of these are like physical based tests, so we're not really doing some enormous room for interpretation. Mm-hmm. But if a researcher or a scientist is like, oh, I got this brand new compound I found in the South American rainforest, and I want to see if it's an analgesic or if it could potentially be used for pain reduction, they're going to do these exact same tests. They're going to do the hot plate test. They're going to get a pig ileum or other animal intestine, see if it reduces contractions. With the cardiovascular tasks that they were talking about at the end with the dogs, that was looking at respiratory depression. And then last two, and I think you mentioned this, but if not, definitely take it from here and explain what that was. But there was one to look at cough suppression. So opiates have always been known to help with coughs. So they're taking care of the pain reception. They're taking care of the internal testin mobility of that smooth muscle. And they also want to know, related to cough suppression, how well does it perform?
Yeah, and I remember on Hamilton's Pharmacopeia, he went to, I think it was maybe Thailand or Malaysia somewhere, and the guy gave him a kratom leaf that had sugar on it, and you wrap the sugar in it, and you eat the leaf, and it's for cough and cold. Uh-huh. Which, codeine, I heard, there's a guy I'm going to interview next week for the podcast, and he said he lives in England now, and they sell codeine over the counter. I guess it's for cough.
Yeah, I mean, I've been to Israel, I've been to Russia, I've been to the Netherlands. Tylenol 3, Canada is another one. Tylenol 3 is acetaminophen mixed with codeine, and is typically an over-the-counter medication in all of those regions. It's only in the U.S. where codeine or Tylenol 3s have to be by prescription, and the stigma of being a pain patient and all that jazz. Like, you just don't see them available, used for if you have sores and a headache, or you have a horrible cough. consuming the classic opiates is definitely a way to get that suppression going.
In this study, they say they use all doses of the compounds tested are expressed in terms of the Freebase. What does that even mean? I mean, I know it's what Richard Pryor did in the 70s.
Well, yeah. So, I mean, that was the example I was going to use in that. It's one that most people know. So the Freebase form of cocaine is crack. And I think that's correct. It could be the other way around. But anyways, it is a different molecular arrangement of the compound of interest, usually with or without a hydroxyl group. The base is generally with the loss of the acid tags, but it's more when there are changes there, and I'm guessing, I'm making an informed guess here, but it's more about the storage, stability, mixability, visibility of the compounds you're trying to get and experiment with. So will we, and I think they mentioned this a little bit, but like they studied the difference between oral administration and, injections into the stomach or the muscle. And so toggling back and forth between the Freebase and the sort of natural state of the drug is totally just like, it's the same drug and it doesn't lead to necessarily different effects, but it can ease different routes of administration.
Now, they say they use Nalorphine in here. Is that kind of like naloxone? Okay.
So, yeah, they were using Nalorphine, which is not a compound that I was familiar with. So, of course, we did go and dig and look at it. Here's what I'll say first and foremost. I do not think it's naloxone. I think which would be like what's in Suboxones now, or the Narcan is just pure naloxone. What Nalorphine looks to be is kind of like Suboxone, in that is a opiate agonist and antagonist mix. So you consume it and you have both a blocker of that drug and that drug in the same thing. It's like a 50-50 mix. That's as best as I can and like really understand why they were doing this. And if I truly had what I think really is going on here is that this naloxone is sort of like an early progenitor of what became naloxone, right? So just back in their day, this was sort of the best to try to induce a withdrawal or to reduce competition with the standard opiates. And I don't think it's really used in much favor or has been used recently. That's a guess, though.
So so they did. I think they did some tests where they compared the effects of codeine, and at one point they said, well, codeine causes emesis and dyspnea in these animals. Mitragynine was devoid of these particular properties. I mean, here's another one. Respiratory depression was more. This is on 156 under cardiovascular and respiratory. It said respiratory depression was more frequent and more marked after codeine than after SKF. That the word for Mitragynine. Yeah.
Yes, I had that highlighted in green. Talk about this because it's like, so we laid out all of the tests that they performed. This is the primary research article. These guys were performing these tests and sending out to the journals. So we're jumping into now the cardiovascular and respiratory effects. So they're looking at is respiration depressed at all? Which, by the way, an overdose, it just means that you stop breathing because the opiates depress your respiratory activity so much. So observations indicate that codeine is more effective than Mitragynine in depressing respiration. So in 1972, they did some studies and they published studies saying, hey guys, it doesn't seem to be this compound or group of compounds that do provide analgesic therapeutic effects also do not cause the respiratory depressions, which is the most dangerous part of the drugs we're currently using. And then you just sort of sit there with your hands up like, well, what are we doing today? Some of the earliest studies here, that was a major conclusion from that,
Yeah, I mean, if they knew this in 1972, my question is, why hasn't a drug company tried to cash in on it like Glaxo SmithKline later? I mean, they have this research available, and it seems to be promising.
So I was thinking that exact same thing, and it's like, gosh, why is no one like pursuing this? I have relationships within people, scientists that are doing pain management, drug development, and this, that, and the other thing. I always say, oh, well, of course they are. Of course somebody's doing it, but it's like, are they? I mean, I don't know really where we're at, and it'd be better to define what a pharmaceutical company is actually looking for too. They want to take mitragynine, they want to modify it a few ways, but not modify its behavior, just attack on a few molecular, atoms here and there. So then it could become patentable. Although this is derived from a natural source, there might be issues around IP. Anyways, I guess the broader point here is like legal issues and potential turnover and IP can sometimes outweigh the fact that it might actually be better and worth exploring in terms of pain management for the populace. It's unfortunate, but they're businesses, and business drives those decisions.
What do you mean by IP?
Intellectual property.
Okay.
Yeah.
So just something happened that made it easier to develop opiates, I guess.
Well, or take mitragynine and then add a hydroxyl group and a weird, tag. And then you can do that. You can patent that. You can't patent just the mitragynine. And then that lasts for so long. It's just, I find it hard to believe that there are not actively people doing this. But when the other papers that we've presented thus far in, late 2000s, like 2010 to now, it seems as if almost none of them, except for one that got us this paper, but are unaware of these papers that are being done. And not, it's a little disingenuous to say no one really has evaluated the effects of the analgesic value, its effects on respiratory system, its ability to suppress coughs on multiple animals at multiple doses, and gotten a relatively steady cross-species profile of what this drug was, and that's exactly what Macko and his colleagues had done in this old paper from 1972. So I don't know. I mean, there are opportunities to help people, and the people that are in pain management, if they're not looking at the mitragynine or Kratom alkaloids, I don't know what they're doing. And maybe the market niche is still open for you and I, Brian, to get a little R&D company going. But
yeah, hey, I'm all about
it. For sure.
I'd probably do the shipping or something. I don't know. It's kind of because, I mean, this company was a drug company that developed drugs. I mean, they had a patent on amphetamine. The one guy went on to develop Lipitor. It's just kind of strange that they wouldn't try to cash in on that.
Yeah, and you get even more cynical too, to say like, well, after they got everybody hooked on their opiates, they also then start sending them constipation meds. So you got a patient that's chronic that has to now buy 2 or 3 pills from us, versus and risk being addiction. And this is where it falls apart, because once a person's addicted, the cost of the insurance companies go way up. But I don't know. It's and I'm not proclaiming that this is what's happening, but it's the classic tension between profits and people. And we can't always assume. I don't know. I'm with you on this, and that this is a good thing and could be a good thing for a lot of people. I don't know why they're not doing it. So I don't talk to them, so I shouldn't really speculate. But it is just frustrating. I guess I'm just sharing in the frustration there.
Yeah, and I mean, you just said about constipation. They actually found no gastrointestinal problems with the charcoal test in rats.
Right.
Some people say that Kratom gives them constipation, but my guess about that is that people are doing toss and wash, and they're just it's just like a physical constipation thing because you're putting cement in your stomach. Because I make tea out of Kratom, and I've never gotten constipated from it, so I don't think it's the Kratom.
And it's all dose dependent for sure. You know, I don't know. We don't have the math on for their studies on ileum movement. We'd have to look at the milligram per kilogram that they gave and then understand if that would be like a high dose or a low dose. But I mean, if you take enough of them, it can be there. for sure. But it is just amazing that it's like, oh, we want the ideal thing to get us out of this opiate crisis would be something that relieves pain well, that doesn't have the side effect of constipation, that doesn't lead to respiratory depression and lethal effects, but also maintains a solid or relatively the same degree of pain relief. And it's like, this paper lays all three of those out over 8 studies that are very strongly pointing that, like, I'm surprised there isn't just this isn't the like patient zero article, and that a whole field of pain management scientists have went from here on to get more of it locked down. So this is an opportunity to do so for any young neuroscientists or psychopharmacologists out there. Experiments need to be done.
Yeah, I wish one of these guys were alive so we could ask them what actually happened. I'm pretty sure they're all dead. I know 2 of them are. I couldn't really. I imagine Macko is too. And the other thing that I found is it said that mitragynine more potent when given orally, and it says there's a possible that the active analgesic moiety may be due to a metabolite of mitragynine. And this study I just blogged about, it came out last year, Andrew Krugel from Columbia University, he had a study that showed that mitragynine actually metabolizes in the liver into 7-hydroxymitragynine, which is the second most abundant but more potent alkaloid in kratom, which is pretty cool. Which is pretty cool. So it's seeing that, and then reading that study, and they did tests on mice, and they did tests on human liver and mouse liver microsomes. And they found that there's an enzyme in the liver that actually oxidizes the mitragynine, changes it into 7-O, and that's why it's more effective as eaten rather than injections.
So first pass metabolism is a really big thing, especially with opiates, right? When it goes to your digestive system that way. So was he suggesting then that the mitragynine that was delivered orally was more potent, but the other, like, codeine that they were using wasn't? Like, could it go through your liver, be more potent, and still as effective as the codeine?
I'm not sure. I'm not sure about as compared to codeine, but it was kind of weird that it metabolizes it, because they said the 7-hydroxy is the stuff that has the more opioid-like effects. And they were saying before that if a kratom extract has a more abundant alkaloid of 7-hydroxy, it might be more potent than the natural leaf. But that study showed that the 7-hydroxy is stable in the liver, while mitragynine, there's a couple of oxygen molecules added to it, and it turns into 7-hydroxy. Yeah. There are always wild enzymatic degradations that happen in the liver that can lead to more dangerous byproducts. It's something that I've had to run into in the cannabis industry, making medicine for sick people, immunocompromised people. Pesticides and these other chemicals are bad, but they can turn into even worse after being exposed to heat and flame. Like, there are very little substances humans consume where they just like burn it right before they take it in. And so there are some real scary combinations that could come out of that. As well, consuming alcohol with acetaminophen is something that's highly recommended against, because the aldehydes that are made due to enzymatic degradation in your liver can really start destroying your liver. Like, they're toxic for your liver in the worst way. So it's certainly something to consider. And it's interesting. It was interesting, and I recently had just tried mitragynine on its own, and I'm wondering, that would have all gotten over hydroxy with this notion. I don't know. I guess I just don't know how if I think that
that's the only thing that's happening, and that's why we're seeing these effects, or if it's a little bit more complicated than that, and the effects are more based on the symphony of those interactions.
Yeah, because I think they also, when they tested, tested the brain tissue, the mice. They found mitragynine as well, but there was a higher amount of 7.0, and then it said some of the 7.0, they gave them just 7.0, and a little bit of that turned back into mitragynine. Now, like, they tested toxicity with this study, with this Macko study, and they gave lots of mitragynine to mice and rats. A mouse was given 920 milligrams per kilogram, and a rat was given 807 milligrams per kilogram. And as far as I can tell, from what I read, I think like humans get an average of like 25 to 50 when they take Kratom. So I don't know if that's equivalent in mice and rats. However, it seems like a large amount of mitragynine, and they observed no toxic effects in mice and rats.
803 just to begin with is a pretty big dose of that, far bigger than what anybody, any products on the market now. So yeah, they were trying to go for the LD50 here, and it said that only produced diarrhea, observed 2 dogs, oral doses, no side effects.
I think the dogs got some more side effects. I think the one dog got clonic convulsion, respiratory depression, panting, prostration after an IV dose. A 31.8, but an 80 milligram per kilogram oral dose did not induce side effects. So I don't know what that. And it showed sinusoidal cellularity in dog livers. So I don't know what that means. Dogs are also allergic to chocolate, so I don't know if dogs are just weird.
It's like a spot on an alcoholic's liver.
Okay.
Yeah, it's interesting. Man, I thought this was cool because like very rarely now are labs afforded the ability to do multiple animal studies with different species of animals. So yes, there are moral reasons now in the US why we're not testing this stuff on dogs. But even like if you're a rodent lab, you're either a mouse lab or you're a rat lab. If you're doing like pigs and ileum, then that's what you're using there. This is nice in that it was before a time where everybody had to specialize so much, and you get your group together, you have the ability to run these tests on different species, and you're finding comparable findings across the board right away. It's I always appreciate reading these scientific studies written, with a typewriter in 72, because it's much less sort of regurgitation of everything that is the modern scientific journal article these days. And it's more it's almost like a lab report that you got an assignment in class, where they're just like very straightforward. This is what we did. This is why we were doing it. Here's what happened. Hooray. It's not mixed in a lot of contemporary, just sort of fluff and posturing, which is real nice to read.
And yeah, I was surprised. Like, they did a rhesus monkey. It said, well, the abstract, I laughed. It was like the first paragraph says it fails to produce excitement in cats, which that was just. I'm like, okay, they found that out. Well, that's good to know. It said it failed to alter the usual hostile behavior in a rhesus monkey. So Kratom does not keep monkeys from being assholes. It was just interesting, just how many animal studies. You said they don't use dogs anymore or monkeys.
In the U.S., I'd be very surprised, except in the biomedical realm, right? If you're at a vet school, I'm sure there's animal husbandry over there. You're working with animals all the time. That doesn't seem that you'd be doing behavioral psychopharm on them and like giving them drugs and observing what happens. But not these type of studies. I mean, it's a problem with a modern, U.S. research enterprise, universities and colleges. And there's a few good anecdote stories from there, but like things have gotten very sterile, very rigid. Things have to be done a certain way. And even though it would be beneficial for a lab to have multiple species to test through, and we did in grad school, we had C. elegans, we had the zebrafish, and then we had mice, but we weren't running the same experiment across all of them. But the bummer of that is it just reduces and minimizes the potential serendipitous discovery that can have when things are sort of, a little loose around the edges. Not everything's super sterile. Most of the great scientific discoveries we've had in the lab are like, it was late at night and dude threw his Petri dishes in the sink and he's like, I'll clean them tomorrow. Comes back the next day and he notices that the mold and the growth on these has been pushed back by this mysterious white substance. What's that? That ended up being penicillin.
Yeah.
So there's also, for a more sort of public health thing, there were, in my pharmacology department at Tulane, they were working on blood flow and heart attacks. They were one of the first to start doing NO2 research. They recognized that when they were blowing smoke in their lab, they were allowed to just smoke cigarettes in their lab. When the smoke hit the stomach muscles of this rat they were performing different dissections on to get some data, they noticed that it started moving faster and beating faster. And long story short there, that was the birth of where we are now today with Viagra. That's how that all started, is because they were smoking in lab and they noticed that effect. And you do a bunch of other research by keeping those animals. So there was this other one that came out of that lab. It was an AIDS HIV drug, but like they did the tests in mice, they did the tests in rats. They wanted to get up to humans, and by the time they got up in humans, it's a potential miracle drug for HIV and AIDS treatment. And what it turned out to be is that the effectiveness of these compounds is so tightly with your internal biome and your gut bacteria that when you're testing in animals where it was sterile, and then go to a human, which is not sterile, the drug doesn't work anymore.
Yeah, that's interesting. Is there anything else that you've observed that, happened in 1972 that wouldn't happen today? I mean, I guess you kind of laid it all out there.
There was. I mean, there's a very big one. And what's nice about this paper is it was not in the world that we have today. And when you read this paper, I mean, it very clearly is saying we have a problem with narcotic analgesics, and we need to find better ones like that reduce constipation, that are not antagonized by Nalorphine, has like reduced overdose risk, and all of that. And so they're starting from that point alone. If you go back to the studies of now, and it'd be great to talk to Dr. Schwager, and the mental health review I think we're going to be doing next week is fantastic, because this example of a paper that didn't have to introduce drug hysteria, panic. And so when you look at the modern papers, it's like opiate epidemic, this is killing people, fear mongering, like baseless claims, and we need to figure this out. Like that, the whole attitude and approach from these guys in 1972 saying, hey, we do need something like this, let's give it a try and see what happens, versus, hey, people are dying, we need to make it a Schedule 1, and we need more science, this, that, and the other thing. Like, it's a whole different narrative that current scientific studies are forced into, that I think we've talked about before. But it's a refreshing thing for me, I think, to understand that these guys were doing it for the purposes of trying to find compounds that would be better without the side effects. And that was sort of it. It wasn't a discussions of addiction and dependence and public health, all of that sort of absent from this. So it's nice to, and it's almost like a vacation to get back into the mindset when they were there. So less politics involved.
Yeah,
certainly.
Yeah, and these guys were they were born in the 20s, so it's probably safe to say that they were probably interested in science as teenagers before even like marijuana might have been outlawed and all that kind of stuff happened. So they probably don't have. And 72, I think, is when Nixon launched a drug war. So they probably didn't even have that whole context for they didn't have the benefit like me of growing up with Nancy Reagan and Just Say No, and your brain on drugs commercials in the 80s.
Right. Yeah, the fight against honest drug science started, and we still deal with it today, for sure.
Yeah.
So yeah, I mean, the only thing in the discussion here, in search for our potent, safe analgesics possessing a profile of qualities which are superior to those of existing agents, Mitragynine exhibited activity in animals which seems to potential usefulness in man. He goes on to say that it differs in other analgesics by alleviating brain threshold in the rat, mouse, and dog with minimal side effects. Mitragynine is an active modality involved in most optimal process. Mitragynine is qualitatively different from the narcotic analgesics, and cats exhibit only a weak stimulation and is more akin to restlessness, where there's no evidence of disorientation or discernible influence on respiration, where Mitragynine was. So, they very clearly laid out, like, these are the problems we're trying to fix, and we've done a set of multiple behavioral tests in multiple animals, and all of them seem to suggest it's effective, and while also doing that analgesic treatment. It's I'm glad that we were able to get this in. The podcast before this, or a few before this, journal clubs, we were looking at broad historical approaches. This is one where we are now in the mindset of the 1970s. And for all the scientists and researchers out there, and myself, it's not always a good thing to filter by year, where it's like, I don't want to read anything that's 2010 or older, right? I want the latest science. I want the best science. Well, it's just shit gets telephoned along the way, and it's always helpful, and it's really exciting and fun to go back and see what people were saying back then. They're generally more clear, less fluff, and just tell you straight what it is. So I really enjoyed this paper.
This is like the last sentence. On the basis of the apparent qualitative differences between mitragynine and the narcotic analgesic agents in these preliminary studies, it can be anticipated that new chemicals whose structural configurations are quite unlike the morphine-type structure may well produce analgesic properties, which are unaccompanied by the limiting side effects of morphine-like drugs.
Mic drop.
Thank you, Dr. Jonathan Cachat. That was the Kratom Science Journal Club, and it's produced by me, Brian Gallagher, for KratomScience.com. The music is Captain Big Wheel. The song is Moonrunner. Like and subscribe to our podcast on any platform, and check out ccvresearch.com to see what Dr. John is up to. And take care.